So what I want to present to you is what's happening with peanuts and aflatoxin in Australia. For the people who are new to peanuts, we grow peanuts not many tons, about 30,000 tons. Compared to China, for example, it is 12 million tons there. So we are a very, very small industry, but we are technically much more advanced industry. 30% or 40% of the dryland crops, they are grown in Burnett region, which is dry land, and that is reducing or decreasing. For the past, at one time they used to go about 12,000 hectares, now it is less than 2,000 hectares. What's the reason? The main reason is aflatoxin and drought. They were getting, even if they are getting the yield, they are not able to avoid the aflatoxin contamination in the product. So the industry is moving north where irrigation is available. So what we produce, 90% of the peanuts are consumed locally, domestically, and very little is exported. In fact, we import peanuts from Argentina or other countries. So to give you a little bit of an example, the research for aflatoxin that we initiated some time ago was driven completely by the research, by the industry. If you look at this here, the prices that the farmers were getting in 1980s until 1992, there was no differentiation in the price. Industry was measuring aflatoxin, but there was no price penalties for the contaminated product. But if you look at this, since 1992, there's a big difference. The contaminated product is bringing less price or less income. So all the industry stakeholders, they have to accept aflatoxin as one of the major constraints for the industry. How that happens is that the industry started penalizing the growers for their aflatoxin contamination. As the contamination increases, the price they get the products drop down even into the half, 50% or more. And these are the classes that peanut company gives. When they analyze the aflatoxin, the products goes into one of these segregations, and then the price is allocated based on the seg value. So a little bit of how this aflatoxin occurs in the peanut kernel is something like that. When the plant is performing well under irrigated conditions, if there is a fungal invasion, there is immune response from the plant. So phytoalexins or antioxidants are produced. So the fungal growth is reduced. So plant can take care of itself if it is once it is healthy. But when the drought hits it, or any disease, for example, which compromises the health of the plant, then it impacts directly on the phytoalexin production and promotes the fungal growth. And if that is combined with the elevated temperatures and reduced water activity, if there is a kernel on the crop, that's where the aflatoxin production occurs. So as far as peanut is concerned, aflatoxin is produced both pre-harvest as well as post-harvest. In pre-harvest, the end of season droughts or soil temperatures or high inoculum in the soil promotes the aflatoxin production. This fungal inoculum in the soil, it's always present there. As Mary said, it is ubiquitous. And we do have both parasiticus and flavus, mostly parasiticus in Australia. Post-harvest contamination can occur right from the field itself, depending upon how the crop is dried in the field before it is thrashed, or when it is taken into the storage, depending on the poor storage conditions. So we looked at three possibilities of minimizing the aflatoxin. One is we looked at the genetics, on-farm management, and post-harvest management. I'll briefly cover each of these. In the genetics, we looked at all the genetic diversity that we have in the peanut germplasm for drought, for foliar diseases or pests, and we tested more than thousands of germplasm lines, we do find variability in the germplasm. We do find some genotypes producing less aflatoxin compared to the others. But the question is how consistent they are is the question. We do find also very consistent responses between the varieties for aflatoxin production. If you look at this graph here, NC7 used to be cultivated, very popular variety in 1990s, but it's only the aflatoxin that it brought it down. It's nowhere grown at the moment. Then we introduced Streeton, which is a lot more drought tolerant compared to NC7. If you look at this graph here, the Streeton always produces less aflatoxin compared to NC7, but it is not acceptable for the industry. Why? Because it is still producing aflatoxin, which is not acceptable for the food standards. So we had to go look for more options. And second thing we looked at is the varietal options. For dryland production, 7 out of 10 years, it's the end of season drought, which is very, very common in South Bennett. So we looked at introducing ultra early lines, which can mature in 100 days or 110 days, compared to what they have been grown 140 days or 150 days. These ultra early lines, they escape the end of the season drought. By that, they also escape the aflatoxin risk. If you look at this photo here, the Streeton compared to a variety called Tag 24, when Streeton is still setting pods, this is nearly complete. So it can complete its life cycle. It's not aflatoxin resistant, but it is escaping the drought. So this has become a breeding strategy now to produce generate ultra early varieties which are suitable for Australian industry. That's happening for the past 5-6 years. Ultra early varieties with characteristics that is required for the peanut industry, they have been released. In our quest for looking at the mechanisms of genotypic differences, we do find a great genetic diversity for relative water content, that is water status of the plant and how it is related to the pod moisture. As I told, moisture content is one of the very important things that contribute to the aflatoxin contamination. If we can keep the water up to a certain level or above a threshold, it can reduce the toxin contamination or toxin production by the fungus. It's only under water stress conditions the plant is producing aflatoxin. Similarly, we looked at the shell wall integrity. That is, integrity means how well the pod is able to keep up its structural integrity of the pod so that it doesn't allow the fungus to penetrate into the soil, into the pod. There is quite a bit of variation there. It is related to the aflatoxin. Another thing is we looked at is the intrinsic capacity of antioxidants, or we did that with the total phenols. So you can still see a relationship. The plants which produce a higher amount of phenols, which consists of a bulk of antioxidants, can tolerate or resist the aflatoxin production. So with the genetic, that's where we stand as far as the genetics is concerned. The things have not moved further anyway, but these are the ideas for future research. And then going on to the on-farm management of it, we need to understand what are the windows in which aflatoxin and Aspergillus growth is being promoted by the crop. Looking at the media here, the Aspergillus flavus parasiticus media, and when it is grown under thermal gradient on a thermal gradient plate in a range of temperatures, you can see here the aflatoxin production occurs in a very, very narrow window. Even if there is fungus there, it is only active in producing aflatoxin between 20 and 35 degrees. And then we did large experiments using detached pots, pods itself, using the peanut pods, different varieties also. The point is that even if the kernel is infected by the fungus, aflatoxin production only occurs in a very narrow range. That's the important message that we need to consider when we develop the practices. And also other more growth chamber studies, they did point out that you should have elevated temperatures, which is 35 to 22 here. Even if the crop is under drought, if it is under cool conditions, there is no aflatoxin production, as it is shown here by the blue bars. Similarly, the crop has to be drought stressed to produce aflatoxin. So these two have to come together. That is, soil moisture and temperatures have to come together to produce aflatoxin. If there is moisture stress without temperature stress, there is no aflatoxin, and vice versa. So the question to the growers is that, okay, how do I know if my crop is under stress? When do we harvest it? If you delay the harvest, you are increasing the risk. So aflatoxin production is enhancing with time. So that is $1 million question for the dryland industry grower. So if you put it into a context of a model here, assuming that the fungus is present all the times here, the three things which control are soil moisture or kernel moisture, or soil temperature, and the substrate, which is kernel. So the aflatoxin is produced only in peanut kernel. It is not produced in peanut leaf or stems. It's only in peanut kernel, not even in the shell. So the substrate has to be there for the fungus. So when the three things come together, that's what we call is aflatoxin risk area. So now we can effectively model this, because these three are, we know about the crop. By measuring the environment, assuming the fungus is always there, by knowing the crop, there is substrate available on the crop, you can nearly put a very simple model in place that the cumulative daily accumulation of these three risk factors is directly proportional to the extent magnitude of the risk. So that's what we did using EPSIM. Probably you would have seen this graph a million times in UQ. This is the EPSIM framework, where you have the crops interacting with the environments here on daily basis. The relationship between the crop growth and environment, they have been worked out for the past few decades, and all those algorithms are sitting in this engine here. So what we did as a part of our project is to introduce a small module called Aflaman into the peanut crop thing. So what it does is that the model simulates the peanut crop on daily basis. It looks at the aflatoxin risk factors. Once the risk is there, it will start a counter. So that's all we are measuring here as a risk index. So retrospectively, when we looked at the model performance, again, is what actually measured by the Peanut Company of Australia in terms of measured positive loads, we do get a reasonable relationship. That means the model is able to predict or simulate the risk of the crop if you provided that with the crop data, soil specific data, and also meteorological data. So the next step is to bring it up, so that to bring the science to the growers. So we developed a web-based aflatoxin monitoring decision support tool. You can still go into this website here to access this model here. All that happens in this framework is that we go and register whoever is interested in monitoring. Those growers will be given a logger, or we'll put in a logger in their paddock to record the soil temperature, air temperature, rain, etc. And those are entered into the computer, and they log a job by through internet into an EPSIM peanut manager, which sits at Toowoomba. The model analyzes the risk, and the risk is given them in one page. So what it is showing is basically, it shows how the soil water is varying in their crop, and looks at if it is below that red line there, which is the threshold, the crop is in risk for water stress. Second thing it looks at is what's the soil temperature is doing. If the soil temperatures are in the risk zone, so both water as well as soil temperatures are favoring the aflatoxin production. So it triggers the aflatoxin risk in a line. If the farmer sees that this line is going up on a weekly basis, then he has to look at what is the forecast for the next week and make a cutting time decision of the crop. So we ran this tool for the past 6 years or 7 years. You can see here that since our project started in 2008, the risk of that is in the industry is drastically reduced. I don't take credit for this, but I think the rainfall also favored. For the past 6 years, the crops had got a little bit more rainfall. And not that the situation will continue for the next season. It doesn't seem to be. But the farmers are aware that this tool is there if they wanted to use it. But even in these years where the rainfall was adequate, they were still looking at this model because they didn't want to harvest the crop in a hurry. If they see that there is no risk, they can leave the crop so that they can get more yield or more seed filling. So they do have interest in it. So another strategy on farm we looked at is the biocontrol strategy of aflatoxin minimization, which actually the principle is that if you enhance the non-toxigenic fungi, their proportion to a desired level, which overwhelm the soil with the non-toxigenic fungus, probably they will competitively inhibit the uptake or infection by the toxinic fungi. Because the non-toxinic fungi has taken the place, there is no aflatoxin production. To put this in the context, in the soils we do have both toxinic as well as non-toxinic. Toxinics happen to be a lot more powerful. They prevail and multiply well in drought conditions and high temperature conditions. But if you infected that with the non-toxinic fungi, there is a chance that you might reduce the infection by the toxinic fungi, so there is no aflatoxin. So we did a widespread trials using this non-toxigenic inoculum, which is applied in a like a cracked barley or cracked rice, where the spores are produced, and it is broadcasted on the soil. And when it is there in the soil, fungus is supposed to multiply, infect the pods, and then overwhelm the pods with the non-toxic fungus infection so that there is no aflatoxin. We did number of trials, but I would like to present only 2 trials here. In the trial 1, we have a fantastic response. For example, the green bars are the inoculum levels, and the red bars is the aflatoxin production. You will see here in the control, the crop is producing about 63 ppb. When 100 kgs of non-toxinic inoculum is applied, the fungal inoculum, the non-toxinic inoculum went up, and also aflatoxin came down compared to the control. If you extend that to 3,000 kilograms or 1,000 kilograms, you have a good response for those non-toxinic fungi in both the varieties. This is VB, and also there is a straighten here. So this is a very good news for this new technology. But in other sites, for example in here, it was totally a reverse case. There is the whole heap of aflatoxin is produced and there is no uptake. So what is causing this fungus not to multiply, and what is causing it to compete with the toxinic fungus, we don't know. That's where this technology is sitting at the moment. So in summary, we did spend a very productive research time in looking at understanding the factors and developing the pre and post harvest management practices and aflatoxin simulation model, and they have been effectively integrated with the industry. This implementation of these practices and pricing incentives to the grower actually has effectively minimized aflatoxin contamination in Australia in peanuts. But as I said again, the rainfall has to cooperate in this section here. If there is no rain, farmers they can't plant peanut anywhere. So when both are going here, we do have tools, we do have practices growers can adapt. We do find the varietal differences, but at the moment they are not enough to eliminate the aflatoxin risk. There is no one genotype in peanut germplasm that is totally resistant to aflatoxin. What do we need to do for future research? These are the areas we think that need to be explored. We do find that conventionally peanut pods is supposed to grow by themselves. There is no xylem connection between the leaf and the pods. So even if the plant water status is good, the pods may not have that status. But our research is showing that there may be some genotypes may have that conductivity link between the plant and the pods. We have not explored that yet. And second thing is pod wall integrity. We did find out that the cracks that develop or the injury that the pod wall gets is the major source of infection by the fungus. How do we maintain the integrity of the pod wall or shell wall to resist infection by the fungus? And third thing is we do find a lot of genotypic traits, including the drought-resistant traits, for contributing to the aflatoxin, but they have not been consistent. Can we pyramid them to put all these things to see if we can minimize that toxin contamination? Other area which I have not presented here is that the biochemical composition of the kernel of the peanut may have something to contribute to resist the aflatoxin synthesis, even if the fungus is present there. The recent area that's coming up is the biotech area, RNAis. There is a couple of research projects going on in US just started, and we haven't explored this area. So I think that's where I leave it.